Dr. Kikueva's,
Can I come
Dr.
up
Kikueva
there on the delta
Kikile's
there, Jake? Go ahead.
Oh, sorry. Dr. Kikueva Kikile's research. So this is actually a link to his dissertation, and then this video on Vimeo here is a link to his doctoral on defense. So if anybody wants to get a little bit more, some in-depth context, then you can definitely go there for those resources. And then, like I said too, right, we have the Office of Hawaiian Affairs, yep, at Papahanaumokuakea, their site, some of the documents and webinars that I was talking about. Right? This is the cultural briefing video, as well as some of the other resources that I was mentioning earlier. And then here we have the not the monument website, in which you can go to explore for yourselves, but that's going to be papahanaumokuakea.gov. And then the other one is going to be oha.org at /.
Sorry, Herc.
Nope.
Can you back up just out of the screen in the lower right corner? There was a stalk with a fuzzy stalk. Right there. Can you zoom in on that?
Would you mind illustrating it?
Yep. Can do. Is it this right
fuzzy
here?
stalk, yeah, that one.
Right.
I need another telestrator back here.
No way. I'm not ceding this power.
Going to be more than $20 for that.
Yeah.
That's it? That's a bargain. But I can get one on Black
No,
Friday.
I said more than $20.
Great. And
All right,
video,
go ahead
you
and push
feel
on
free
in
to
there,
put
Jeff.
whatever you like on channel 3.
So this is another octocoral that I don't know if folks have seen on previous watches, but it looks like a stoloniferan. And sometimes these are much harder to see because they don't produce the skeleton that they're on. Looks like a sponge or coral skeleton.
Yeah.
But nonetheless, it's a component of biodiversity that's often overlooked. And we had sampled something very similar on another seamount, and it's tough to tell if this is the exact.
The different coloration
thing.
at the base of some of those polyps, is that?
Yeah, sometimes these stoloniferans can be pale and translucent like this, or they can be, some have a purple coloration, looks quite nice. But yeah, this is a really good example of some of the hidden biodiversity that we
Oh, right there, Jeff, please.
overlook during these types of surveys. But great, thank you for the zoom.
How are we doing on our latest ship's move, Rennie?
I just called in another one.
Okay.
So we were kind of pseudo-continuous.
Yeah, still looks promising.
I'll just keep doing that. Just let me know if you want to
Okay.
pause. But we'll have a little bit of swing in Argus, but it won't be as drastic as when we were almost double the depth.
Two requests, Jeff. Would it be possible to return to the high pack on channel 3? And can we make the telestrator visible for the public to follow along?
I just put the telestrator out, and oh, wrong one. Hang on.
You're so far ahead. Look at that
Too many
swimming brightway
things.
there.
Yep.
Wow. Did we miss that? Lots of gratitude for your context, Kalame. Thank you so much for sharing that and the resources.
No problem.
We have
Thank you
folks
so much.
watching from Kauai. Someone's been watching this entire dive. Wow.
Wow, nice.
Mahalo for tuning in, gang. We hope that you guys can, especially if you're here in Hawaii, where the expedition is taking place.
How's that?
We encourage you guys to please share these resources out. I do realize I'm very privileged as a Hawaiian to be here in my capacity, but we hope to be able to create more opportunities for access for our Hawaiian communities, especially those that have direct genealogical and cultural ties to the monument as well. believe this is OET's first time partnering with the community, and so I hope that I'm doing my best to represent you guys out there, and if I'm not, please let me know. But like I said, we look forward to having more of our communities out here. So please pass on this information. The Papahanaumoku, the monument website, the OHA website, and the OET website. Yeah, this is a good opportunity for our younger viewers. Well, I mean, for all of our viewers to be getting involved in these pathways. You don't. I mean, we might be able to share a little bit later, but you don't need to have an advanced college degree to join us. Just come and join as an intern. Look at the different various career pathways that might interest you. I think that this could open up a lot of doors for everyone in the community. So please, please, please share this out. We also have some limited time slots left for our free ship to shore interactions. So if you or anybody that you know in the community has a school that might be interested in the multidisciplinary work that's happening out here, please head to the website and sign up.
Jess, do you mind turning, or a video, I'm not sure who does it, the lasers, please?
I got it.
It's Jake. There we go. Thank you.
Thanks, Jake.
Yep, no problem.
Yeah, so now you can maybe make out those two green dots. And what does the laser tell us, Beth?
So the two lasers are aligned to be, those 2 points to be 10 centimeters apart. So that helps us calibrate the scale of what we're looking at, especially if we try to request a rock that may be way too big. This helps us see that, but also helps us see the scale for the animals that we're viewing.
Now we just need a underwater scale.
Right, yeah.
Working on that
I clearly cannot calibrate
technology. Yeah,
the weight that Hercules is capable of lifting, because that rock last night was...
about 51 pounds.
51 pounds, yeah. That was, I think, our biggest rock that we've collected so far.
Yep.
I still don't know how Dan got it into the bio box, or the starboard box. The view in the Argus cam, channel 2, is just really spectacular. See how much density of sponges are up ahead of us.
Yes, that's channel 2 if you're watching on the full screen channel 1, or if you switch to your quad view, you can look in the upper right-hand corner and see the Argus view. Quad is kind of fun. You get some information from both Hercules, Argus, and right now we have our HIPAC or mapping. Actually, I think we have essentially, you can see the ship in relation to Argus and Hercules overlaid on our most recent map that was created of this region.
Can we get a partial zoom right here, Jess?
Sure, on the coral.
Yeah.
Sure thing.
End of that looks different than what I have seen so far. Not that I am a coral expert, but it attracted my attention.
Right, I wonder if that's a farreid sponge.
All right, Jeff, go ahead and do a partial there.
Those barnacles, or what are they?
So this is going to be
Barnacles, yeah.
bamboo coral. This particular colony, it looks like it has one very low branch.
Mm-hmm.
Seems like it branches on the node, but it's tough to tell.
You can push in a little more
You
if
would
you
have
want
to
there,
zoom,
Jeff.
yeah. You would have to zoom closer to the base there. But what's on the
Are those
end of that
barnacles
are
on
those
it?
barnacles, yeah.
Oh, okay.
Okay, that's why it looks
This one
different.
seems like it's nodal branching. The node, the most basal node appears to be very heavily calcified,
Mm-hmm.
and that sometimes happens with these older bamboo colonies. They overgrow their nodes.
So you can see those are actually arthropods, those barnacles related to crabs and lobsters, and they are stalked, and you can actually see their little arthropod legs, those little cirri out there for feeding things.
Yep.
Thank you, Jess.
Yeah, sure.
Thanks, video.
While we're here, let's also take a look at the sponge.
Sure.
Yeah.
It's a very spongy sponge.
I can't quite tell if this is one of these sponges we saw earlier that's just concave but in the upslope direction, or if it's actually something different.
The base of it's fantastic.
Yeah, all those glass fibers.
So cool.
It's definitely got a good strategy of attaching to two different rocks, since the bottom one seems like it might not be as stable.
That's planning. Sure.
All right.
All right, thank you, Jess.
Yeah, sure thing.
Yeah, that you can see now coming into view in center screen, there's a base of a sponge that just got totally ripped off.
Mm-hmm.
You can see the edges of where the sponge used to be. And often you see these traces of life, like these bases that belong to these corals or these bases that belong to the sponges, that gives you an indication of how long something's been in place.
All right. I'll boogie ahead a little bit here. It's just such a nice little outcrop of creatures.
Yeah, you could easily spend all day here looking at just maybe a few coral colonies, because what we are overlooking a little bit is all of the associations that these corals are having with other invertebrates. We saw a couple of instances of straight predation earlier during breakfast. I noticed a massive sea star grazing down one of these bamboos.
Beautiful.
But you also have this, like this Walteria sponge here with all the long fibers coming out that has lots and lots and lots of small little crustaceans in and around it.
Yeah.
Those other things like brittle stars will often make those organisms their home.
Yeah, we're we have such an abundance here that we're not spending as much time with each individual as we were in the deeper depths to see some of those relationships. But we can also choose to do so. Maybe, Rennie, before calling in the next ship's move, we'll settle out and spend a little bit of time investigating those things.
Sounds great. Yeah, we just, ship just stopped moving, so
okay.
as soon as Argus
a meter tall, that thing.
its movement, we'll be in good shape.
So, Steve, I'll let you call out which one you'd like to zoom in on and spend a little time with.
I've been starting to see some some predation, like in the background here at 12 o'clock. There's a small goniasterid that's preying on a bamboo coral, but those are actually quite common here. We also have a bunch of these Paragorgia colonies that are being parasitized by zoanthids. What else? Some of these Walteria sponges, like this one up here, might be a good one to look at.
This one here?
Yeah, the left, so like 12 o'clock now.
Okay.
This long columnar,
yeah, can
big
we spend
branches.
a little bit of
Those
time with
things
that,
are
Jess?
often loaded with
Sure thing.
associates. Yeah.
Go right.
There's a free living brittle star down there. That's really unusual.
Go ahead,
To
Jeff.
see. For looks like an asteroschematid, but it could be something else.
Looks like there's a tiny little mushroom coral down there.
Yep. Yeah, so we've got some, we'll just work our way up the colony and see what we see. But it looks like there's a squat lobster there. It looks like possibly a lithodid, or no, not a squat lobster, but a crab, lithodid, very large rostrum.
Can we get the lasers off, Jake?
As well as a bunch of other brittle stars, hydroids also.
Where do you see the
crab?
Oh, yeah, there it is.
Yeah, the crab's white in the lower left, and then hydroids off to the left, and then the brittle stars on top.
Can you circle the lithodid?
Yeah, that
this is what you're discussing.
Oh,
a little tiny one.
And you can tell that because of the number of legs, is that right?
A squat lobster and a lithodid will have the same number of legs. Well, actually, any closer in there? I may have to update my identification on that. But it just struck me with the spines as they are that it was more like a very, very small lithodid and not a squat lobster.
Come a little wide there, Jeff. Thank you. All right, full wide, please.
But yeah, so that, you know, these kinds of observations are valuable to our community, and the more detail we can get and the more time we can spend,
I think we'll need to catch up a little bit.
we can send these images off to specialists, and they can give
Roger.
us some really reasonably good identifications from
Ready.
imagery based on what they have experienced from other cruises in this area, for example, or their specialized taxonomic knowledge.
I love this question because it's coming in as we're watching some beautiful coral, but there's actually a question for you, Beth, about your jelly bean rock that was collected last night.
Oh,
Do you have an update for us on
wow, we
what
do
that looked
have
like in person?
someone who's been watching for a long time.
Yeah.
Yeah, so on our dive that ended about a little less than 12 hours ago, we observed an area that there was somewhat minimal sediment cover compared to the surrounding, and a very shiny looking rock in that vicinity where what looked like the manganese crust that had formed on the rocks, it was just very, very shiny. And Jess was able to deftly recover one of those samples, and we processed it in the lab last night. So the top part was indeed very shiny, about a ½ centimeter to a centimeter thick crust over a relatively unaltered basalt, although the bottom part had kind of an Oreo cookie crumble feel to it. So we've definitely got precipitation of the oxides coming from the water as well as from the sediments below. Yeah, and it was one of the easiest samples I've processed. The crust came right off with minimal brute force in my rock smashing box.
And then the follow-up question was, how much did it weigh?
Oh, we didn't weigh that sample.
Okay.
You know, it probably weighed 1 to 2 pounds. It was pretty small. So it was not a contender for heavyweight champ of the dive. I think 51 takes the cake. We get another 50 sampler in there, you take 100 pounds. Yeah.
Steve, was that the heaviest sample you've ever processed?
Steve stepped out of the van for just a moment. I don't know that they have processed it yet. There's still a few left from last night's dive that still need to be photographed. But it will definitely need like a very strong box for shipping.
I thought I heard him say this morning that one we're not touching, we're boxing it up and sending it.
Yeah, exactly. So those, but even just getting it onto the counter to photograph it is no small feat.
Yeah, thanks so much for your comments and your observations and your questions. Keep them coming. We're so excited to be here with you in a daytime watch, especially if you're from Hawaii. Feel free to throw in your comments, questions, where you're from in Hawaii. How did you hear about us? It's a beautiful area of the Don Quixote Seamount. We're hovering at around 1,800 meters, which is not shallow, but is our shallowest dive yet in this Lu'uaea Ahiki Ikapapaku expedition. Our expedition named for going up and down to the seafloor.
We've definitely been doing a lot of up and down these last few days with relatively short dives and
quick turnarounds.
Quick turnarounds. So trying to show you as much of the seafloor as we can in different places. Especially we have we anticipate that the swells are going to be increasing to the point where we're not going to be able to safely launch and recover. So we're anticipating heading a bit south after the end of this dive to get to a more suitable spot and do some mapping, fill in some gaps of understanding of the seafloor in this region. So Beth, yes, we're getting to a more stationary point here. Is there anything that you want to sample, image, or shall we keep continuing? Yeah, looks like Steve's coming back in. Let's spend a little bit of time looking at some of these individuals. And if I could also ask video to put up the sonar image in channel 3, just so our audience can see kind of the steepness of the terrain that we're looking at as we're trying to decide where we might go next. And Rennie, can you tell our audience what they're looking at there in Channel 3?
Sure. So we have two circles. One is a scanning sonar on Argus, that's on the left. And the one on the right is on Hercules. Argus is mounted on the bottom and Herc is mounted on the top. The red rings on the Argus one on the left are 20-meter divisions. And the one on the right for Herc is 10-meter divisions. And they are both vehicle relative, so the top of the screen is always the way that the vehicle is facing. It's not north, it is the way that the vehicle is facing. So we have Argus facing due south right now, but we're moving at 135. So you can think about if north is the way we're facing, and that's the top of the screen, then something like northwest is the way that we're moving. And in that direction, you'll see a bright return. So the color ramp is from blue to yellow. So that yellow blob we see, we're kind of headed towards that. And that is indicative of a higher slope, possibly a wall if it's strong return. And so Jake's over there navigating the depth that we keep Argus at, is to not run into anything, while also allowing for Jess to have enough leash to look at stuff. So if we're starting to get pulled off the wall, we may be too high, and if we start to have Argus too close to the wall, then we'll need to come up a bit. And then you can see in Jess's the Hercules one, we're right up against a wall, and you can see that in the camera as well.
Yeah, that's no surprise.
Steve, it seems like a lot of these sponge prefer like an ear shape morphology as opposed to the lobed or the mushroom that we've been seeing. Is that
just while we're here, can we just get a partial zoom on this one?
Yeah, sure thing.
I think that would be a great Chris question, because I'm not too sure about how some of the hydrodynamics work around some of these sponges, but I'm imagining that the sponges can increase their surface area to volume by creating these concavities. I don't know why that would be useful for the sponges, but usually when the question arises, why is something shaped the way it is, the answer is usually surface area to volume. Try and expand the surface area for, I don't know, anything from gas exchange to particle capture, food capture. So yeah, definitely. With corals, it's interesting. A lot of the colony morphologies are associated with trying to maximize particle capture in high-speed flows, high-speed turbulent flows.
If you could pan to the left a little bit. Sorry, Steve, to cut you off.
No, that's okay.
It looked like one of those.
Keep going.
I was about to make the mistake of talking about something that I probably don't have a lot of expertise on.
Yeah, this looked a little bit different. If we could get a partial zoom on that.
Yep.
Yeah, sure thing. Let's see if we can get a good shot of that.
I think this is going to be a farade, but let's take a look.
Can you say more about a farade?
Farade sponge, yeah. Type of glass
All right,
sponge.
Jeff, go ahead and push on in.
In the family Farreidae.
And can we get the lasers off when we get zoomed in?
Were you looking at that rock underneath? So
no, I just wanted to look at the sponge because it was different than what I had seen yet.
I'm going to come a little wide there, and I'll park it right here.
That rock looks like it would just fall apart if we picked it up.
All right, go ahead there.
Yeah. So I'm very interested, as someone who doesn't know, if as we move up this seamount, if we're moving into different geology than we have seen at deeper depths. Yeah. I mean, the crust is still crust, right? But what's under the crust can change.
Well, and as we get to different depths, the eruptive events also might be different. Thanks, Jess, you can pull out on that.
Looks like there was a tiny crab on there,
yeah.
sort of maybe a squat lobster.
Okay, I don't think we'll want to do any sampling right here, so Rennie, if we want to go ahead and get another ship's move in.
Sure. I think 135 is working for us. Bridge now.
Oh, there's a very nice sparse branching bamboo.
One more step, 50 meters, 135. Thank you.
That was one of these high priority collections from some of our scientists ashore. We did manage to get a piece earlier in the dive to help with that research and understanding some of the diversity and morphology variations in bamboo corals.
Jeff, you want to do a partial zoom on this guy?
Some
like half of
closed
them are
and some-
attracted. Yeah. Jinx there, Rennie.
Yeah. Yours sounded much better.
I like closed. That's a good word.
Bigger words.
Great.
Did you say this species was a high priority, Steve?
It something that looked like this. Yeah. And so this particular image that was sent to us was from Johnston Atoll, a little bit further.
Oh, right there, Jeff, please.
South and west, I think. And so they were very curious if we saw this again, we could sample a piece of it.
Yeah, we got that on an earlier dive, right?
Earlier,
or was that early on this
earlier
dive?
during this dive, yeah.
Okay. So it's safely tucked away in our bio box.
Yes.
All right, so we'll got a ship's move going in. We're going to continue our trajectory southeast and slowly climbing as we do. We're at roughly 1,850 meters, which is the shallowest we've been on the seafloor, I think, on this dive series.
Shallow being a very relative term, right?
Yes. Yes.
Seems to be paying off, though. This depth seems, well, like Steve was saying, it's either, it could be the currents, prevailing currents, because we're on a different aspect of the slope, or maybe it's the depth, or maybe it's just that particular location.
Jake, could we get the lasers back on, please?
Sure.
Thank you.
Sorry.
No worries.
Another good source of some of the hidden biodiversity are these puffball Chrysogorgia colonies. Oftentimes,
Like this right here?
Yeah.
Okay, for our audience at home.
Chrysogorgia colonies, yeah. Will often have sometimes shrimp, but usually squat lobsters. Sometimes one, sometimes more, depending on the size of the colony.
And do a quick partial there, Jeff.
Survey says.
I see one in the colony above. I don't know about this one.
Yeah.
Oh yeah, a little
Little
Maybe
tiny
a little
one.
Yeah.
Little tiny one right there. Yeah.
And what's actually really interesting is I'm a part of a project working on coral associate relationships led by a scientist at Woods Hole Oceanographic Institution. And we've been trying to identify patterns among associate coral relationships. And from further south, from the equatorial Central Pacific, and what we've found is that depending on the depth, actually, the strength of the association can be quite strong, or associates may choose other types of hosts to live on.
135.
Typically, the deeper you go, the stronger the association. So
you can almost
the deeper you go, the more you see those two associations together. So species A with species A coral, and then the shallower you go, the more flexible the associates tend to be with their chosen host.
That's interesting. You'd almost expect the opposite, that the deeper down you can't be as picky about where you associate.
Yeah, there's less choice for sure in some cases. But sometimes those associations are actually really important diagnostic characteristics for identifying corals too. Species like Metallogorgia, this Chrysogorgia octocoral, very fine polyps, but a very long, almost like a parasol-like tuft of polyps on top of a very long stalk. Has a very strong association with one particular species of brittle star called Ophiocreas. And it's only ever been of that particular species, Metallogorgia melanotrichos, has ever been associated with that one species of brittle star. And it's actually been very helpful for us because we've seen other things that look like Metallogorgia, but if it doesn't have that Ophiocreas brittle star, it's probably not the same species of Metallogorgia. That's just a little bit of a teaser. That's data that hasn't been published yet.
I'm going to scoop you.
We have a former science communication fellow checking in from Coventry High School. Hi, Tiffany. Thanks for bringing your class along for the exploration. For those of you just tuning in, we are, I don't know, at least over halfway through this particular dive. We're carefully watching the conditions at the surface in order to make sure that we can safely recover, hopefully by around 4 p.m. Hawaii Standard Time today. We're here on the Don Quixote Seamount, which is the same seamount that we dove yesterday, just a different area of that seamount. We're here on the north aspect. And seeing a lot of life in this region. What is that purple?
Number I've seen.
Oh, what is that?
Oh, Holothurian.
Oh, I changed my telestrator. Go back. Can we get a partial zoom on this, Jess?
A little sea cucumber.
Yeah, it looks like a sea cucumber. Can we get the lasers off, Jake?
Yep.
Yeah, how these sea cucumbers are deposit feeders. They're feeding on the sediment, ingesting sediment, which is why
Thanks, sorry.
you typically
Jeff, go
find,
ahead, push on in there, please.
typically find these associations of sea cucumbers on soft sediment, but they can also be found in the hard bottom too.
Interesting. You can actually see kind of the spines that puts them in the echinoderm family there.
Yeah.
Seems so soft, but still spiny.
Yep.
Yeah. They're quite fragile.
Are we looking at the oropodia there in the front? Is that the?
Yeah, I think that's the oral side facing us.
Mopping up the
Mouth
seafloor,
parts.
recycling out the other end.
Yep.
Oh yeah.
Recycling is a good word. Yeah.
Very cool.
All right.
Okay, we
Oh,
can.
right there, please.
Yeah, thank you.
Yeah, that's very, all Therians are very curious creatures, aren't they? Spend your whole life vacuuming up the seafloor.
Yeah. I like when they go up, some of them go up in the water column, do a little dance.
Mm-hmm.
Uh-huh.
They're very
But it's like,
pretty swimmers.
well, why? Because there's sediment. It's usually like in this mud flats. It's like, was that patch wasn't good enough for you?
Here's another one of those.
Yeah, well, Walteria
Yeah.
with rhinoids and brittle stars. Yeah, you can see
One up
the
there,
bottom
Dr.
half
G.
of those sponges typically doesn't have a lot of associates, but as you move up higher, you can tell the things that are feeding in the flow. The suspension feeders and filter feeders are much higher, so it gives them a bit of an advantage.
You may have said this before, Steve, but is there any benefit to the host to have those associates on it?
It depends on the association. There has been some suggestion that brittle stars, for example, may help keep the coral clean of potential contaminants, you know, the action, the physical abrasive action of moving perhaps parasites or removing deep sea mucus, gunk, these types of things that may accumulate might be beneficial to a coral. Sponges, I'm not so sure about. I would defer to Chris on that. But yeah, there's some hypothesis that could be, they could be benefiting from that. Yeah, just the presence of the brittle star.
Oh, it looks like a little fish off to our right.
Continue up here. I'll add another ship move.
Roger.
So in addition to imaging all this fantastic life, corals and sponges and their friends, one of the other goals of this expedition is to try to understand the age and origin of these seamounts. So we're also on the hunt for rocks that'll be suitable for that purpose. Hold on one second.
It's quite dry in the control van today.
Saw a pin cushion.
Sorry?
Pin cushion.
Oh.
Why can't I think of the word? I'm
The
thinking
chynoderm?
orchid, but that's not it. The
Are you talking about an urchin?
urchin. I don't know why orchid came in my mind. There was a little one, a white one.
Yeah, I think I saw one just a little bit below us too.
Yeah, that was the one. Yeah.
Okay, yeah.
Yeah. So that might have been a pancake urchin. They're also deposit feeders, but occasionally you find them on hard substrate. Yeah, the holothurians. That's the thought I was thinking about. It seems that for the sea cucumbers, which we call holothurians.
Oh, can we look up this yellow feature?
Oh, yeah, sure.
They seem to use the swimming action really not as a mode of transport per se, but rather to get up off the bottom so the currents can carry them to another place. They seem to have a very strong ability to sense pressure waves. Yeah, so you'll notice that when Herc was getting closer, it started to lift up part of its body and getting ready to lift off, basically.
Interesting.
All right, Jeff, go ahead and push on in there.
I'm wondering if that's some sort of evolutionary adaptation that allows them to, move far distances without spending a lot of energy.
Yeah. Steve, what are we looking at here?
So this is a type of coral that we've seen relatives of
Can we turn
on
off
this
the
dive
lasers,
so
please,
far.
Jake? Yeah, go ahead.
Yeah, so we're looking at a couple of things here. So the base skeleton is probably not created by this coral. This is a zoanthid, and the base is usually some sort of bamboo coral. I bet if you were to peel away some of that yellow, in fact, you can actually see some nodes towards the base of the coral. The original coral was a bamboo coral, which has then been overgrown, parasitized and overgrown by this zoanthid. And I'm wondering if this actually could be, I think we're a little deep, but I wonder if this could be
come a little wide, please.
One of the gold corals, Hawaiian gold corals.
We need to
Kula mana, but I think we're a little bit deep for that, and I would have to check my depth ranges.
Yeah.
But it certainly has the appearance of such.
Okay, we can keep going, Jess. Thank you.
Roger that.
So really fantastic density up here. We're at about 1,840 meters, a little bit shallower. The view in HYPACK seems to indicate maybe we're coming up on a flatter area, but to be determined, looking at the sonar.
Yeah, it looks like there's a bright feature in Argus, so
yeah,
kind of like
a
wall,
pinnacle.
Yeah, a pinnacle of some kind, and then less slope here at the top.
Yeah.
But wow, the bamboo.
30 meters out.
There's a ton of
corals,
bamboos. Yeah.
Yeah, bamboo corals are one of these groups that have been very successful in radiating in the deep sea. Lots of species. There are shallow water bamboos, but they're pretty rare. I think if I recall, the original bamboos used to belong to one
Wow, look at how big
family
the
called the
sponge is.
Isididae.
That's huge.
That's recently been split up into several different families, and I think the type of that family was actually a shallow water organism called Isis.
Jay, could we get the lasers
where
on
you
again,
get
please?
Isididae from.
Sure thing.
There we go.
It's quite a fan there.
That's a good sized colony. I don't think this is a bamboo though. This is actually something else.
Can we get a partial zoom on that, Jess?
Go
It's
ahead.
a large fan. Lots of commensals there.
Yeah.
This might be one of two genera of primnoids. A lot of brittle stars there you can see.
Starting to see some like yellow coloration on the tips of some of those stars.
Yeah, that's probably fouling by other organisms like hydroids. You can see a bunch of hydroid stalks on there.
Mm-hmm. Yeah, right.
Any
in here,
other,
some
any
hydrates.
ability to push in? Yeah, great. So it's a primnoid of some type. The polyps appear to be orienting. So if you were to close, if the colony was to close its polyps, so to speak, it looks like the polyps would orient down axis. That's what sometimes people call polarization. So they're polarized down axis when they retract. And that's indicative of a couple of different genera.
Come a little wide there, please.
But there's also exceptions to the rule. But I'm wondering if this one could actually be Paracalyptrophora. It's one that typically starts out with lyrate branching, which this one does.
You're going to come
And then it
full wide?
follows with dichotomous branching in some cases. So these are kind of characteristics we're using to narrow down our identification based on what we know from imagery of the seafloor and specimens that have been collected. The size of the polyps is also quite important. There's a similar genus called Calyptrophora that has upward oriented polyps. So one of the rules, but of course there are exceptions to any good rule, is that Calyptrophora has upward oriented polyps when they're retracted, and Paracalyptrophora has downward facing polyps, but the genus Narella also has downward and upward facing polyps. There's a couple of different clades that have different characteristics.
Question for you, Steve, about that golden coral we saw a moment ago.
Yeah.
Or a couple moments ago.
Why
is it called golden coral when it is a zoanthid?
It's a zoanthid, yeah.
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 What's the relationship between zoanthids and corals?
Yeah, yeah.
Oh, can we image this?
If I didn't make
little
that clear out, I can clarify that. But yeah, the bamboo coral, so these gold corals often settle on bamboo coral skeletons, but they are zoanthids. They're a completely different group of cnidarian. And the gold corals actually have the ability to secrete their own skeleton on top of the bamboo
Go
coral.
ahead, Jeff.
Some of them are exceptionally long-lived,
I think
thousands of years old.
Question was just kind of the confusion about the naming. But
Yep.
Yeah. Cool fish. Potentially a little type of
There's a
cuskeel.
little rat tail grenadier.
Yeah.
Kind of interesting, Steve.
Is it
I'm actually
grenadier?
looking at the scientific name of you, and it actually looks Hawaiian.
What was that?
Sorry, I'm just looking at the Hawaiian gold coral.
Yeah.
Kulamana.
Yeah, that's the
Haumea.
genus name, yeah. It was described from 2013, so not too long ago. In the.
That's very interesting.
All right, full wide there, please.
Yeah. Thank you, Jess. And
Yeah, nice.
Is there any context to that? I've never really looked into this group a lot. It's not one that I often work with.
I'm just looking at the name, and it looks Hawaiian. So
yeah.
Kula is actually gold, and then Mana is like basically branching.
We
and
can
stuff
turn
like
the
that.
gain up a
And
little
then
on Harvest here.
Yeah, we actually just got up high enough, so it's flattened out, but I can, yeah.
Haumea is actually another form of papa. So she's like one of the oldest, like most revered goddesses in the pantheon.
Your microphone got adjusted again.
Right.
Just want to make sure it's parallel to your mouth.
There you go. Is that good?
Thanks, Rennie.
Yep.
Sorry, yeah. So we just talking about just made a little bit of an observation. Thanks to Steve and we were looking at the Hawaiian gold corals, but the name it looks Hawaiian. So kula mana mana. So kula is gold, and then mana mana is basically like branches and stuff like that. So for instance, your hand is your lima, right? But your fingers are your mana mana lima and stuff like that. And then haumea, I mean, I'm guessing that if that's an association to the goddess Haumea, she's actually another form of Papahānaumoku, as it's seen in the name for the monument. And then if it's Haumea, then I'm guessing that it has to do with the process of not colonizing, or basically literally giving birth. So it's like the birthing gold coral.
Yeah.
From what I would understand.
This view is amazing.
I know. Look at all.
Yeah.
Should we try to get an eDNA?
Yeah, we can do. Let's see.
There's an Iridogorgia back there.
Yeah, I'm just trying to space out the eDNAs.
Okay.
So we can take one and then have some for background.
Sure. Maybe I can try to collect a rock around here.
Yeah. That would be also good.
Yeah,
Yeah,
looks
the
like
ship
there's another
is-
one of those.
I want to save one for when we leave bottom, so that we have like a background sample.
Okay. Rennie, what were you saying?
Yeah, the ship's move is complete. So Argus has a little bit of swing. It looks like we're at this kind of flattened area, less slope up here. It's still a little bit upward. But yeah, we're good to settle here if that's what you guys want to do.
Okay, I'm going to scan the horizon here for some suitable sampling areas.
Roger that.
My suspicion on the gold coral seems to be that I had thought it was a little bit deep to be seeing that particular genus, and we may actually be a little bit deep, but that doesn't mean it's not. It couldn't be. There's the family that coral belongs to is the Parazoanthidae, and there's other zoanthids that look very similar to that are within this depth range. So yeah, without a specimen, we really can't tell if it's the same or not. Since these zoanthids, a lot of their diagnostic characteristics are internal.
Beth, what type of rock are you going to be looking for? These all look kind of jagged. Are you looking for more lobby, or what you looking for?
Yeah, I'm kind of scanning for both at this point. For the jagged one, like there are a lot of jagged ones here, but I want to see if there's some that look like they're freshly broken. And also maybe something that's altered. So I'm not quite sure yet. Just kind of scanning the horizon.
Roger that.
Some big stuff behind
Where's
you.
the
You can see in the
chonoclomps to
Niskin
tell me which rock to get?
Forest of giants.
Yeah, 2 giant somethings.
Are we
But not rocks.
fully zoomed out?
I believe so, yes.
Okay, thank you.
Definitely take a look at the Argus view if you're not in your quad view of at nautiluslive.org. You can see that we are just surrounded by these coral and sponge. I say we're on the ship, but Argus is surrounded. I'm sorry, and Hercules.
Those are a little big.
This may be a Chris question, Steve, but it seems like coral show different pigmentations, but for the sponge, white seems to be the color of choice. Any thoughts on that?
Yeah, there's a fair diversity of coral, of sponge colors. For the glass sponges, which most of these are, in fact, all of them are right here. Yeah, it's usually either white, translucent, or some form of opaque. And there's, occasionally, like Chris showed us earlier, that there's this one cotton candy sponge that was actually quite dark yellow. I'm not sure if that was something innate to the sponge or if it was some sort of something in the sponge that was growing. tough to tell. But yeah, there's an energetic cost to everything that an organism does. And for example, producing pigment, if you live in the deep sea, can be energetically costly. Presumably there's takes some sort of process, metabolic process to create the pigment. If you're that animal, and then if you're living in a food poor environment like the deep sea here, it can potentially be stressful. So a lot of things in the deep ocean tend to either use pigments that have been bestowed on them through evolutionary processes. So their ancestors had this ability to produce this color, and they've just kept doing it, or they produce these pigments to serve some function. Like the color red often is pretty characteristic in the deep sea because it's very quickly attenuated and appears invisible to things that use the visible light spectrum.
Sorry, Steve. So Beth, are you seeing anything that you want to collect here, or?
Nothing immediately is striking out to me, but I'm still looking.
Roger.
Steve, we're just poking in on the digital stills cam. Is that set to
Yeah, it's capturing right now.
Okay. It looks like, yeah, it focuses. Oh, curious, have you been seeing that shroud?
Yes, that's always been there.
Interesting. I don't remember that before. It might be.
Yeah, so one way to get around that is to zoom in.
Yeah.
But it has to be in manual mode for that.
Right.
The other way is to just we can clip it out post-process. Kind of as a.
Has it always been that way, or was it mounted closer to the lens?
It was that way last year.
Huh.
And if there's a way to remove that, I don't know how.
Yeah.
It's always been a feature of the images we've taken this year.
Okay, yeah, just checking.
Whoa, check that out.
Yeah, if someone, if anyone wants to operate the stills camera, you can take control of that computer. Some of the images, let me see.
Jake, you want to give me a little leash here?
Taken about.
Sure
Yeah,
thing.
some of the images are actually pretty decent on auto mode.
Yeah, it looks like it was looking blurry, but then it looks like when it actually takes the photo, it focuses, and then
yeah, it's autofocus.
And we change our depth, and it unfocuses again.
Yeah, the photos that were taken yesterday were quite nice too. Stephen had taken control of the camera and was messing around with some of the buttons and got it to take some really nice photos.
That's cheating if Stephen or Aaron is on it.
I think he's competing for which of the video engineers has the ability to operate the most cameras.
Yeah.
Can you tell us about what camera you're talking about? We don't have access to that on broadcast, of course, but
yeah, we have a camera on loan to us from the Thurber lab at Oregon State University.
That's good there.
And this camera is a very nice still camera that we can operate remotely, or we can set it to take images across an interval of time. In this case, we're using about 30 seconds. And every 30 seconds, it takes an image, and it produces a very nice complementary image to what Herc's seeing through the HD camera.
It's mounted on the light rack on the front of Hercules.
Jess, if we could sit down and maybe spend some time looking at these rocks.
Yeah. Yes. Let's do it.
Hopefully there's not something right underneath us to prevent us from sitting down.
Yeah, good luck.
Jake, can you come down on the delta a little?
Come down.
Yeah, that's a very nice bamboo coral. Sorry, I was distracted. I didn't get a chance to take a look at it properly.
Can we get the lasers back on when you have a moment?
Yeah. But has that camera always been oriented on the light bar, or did we have it in a different place last year?
We had it in different places, but last year's primarily on the light bar.
Oh, hey, look in Argus cam. There's a pelagic holothurian.
Yeah.
Oh, wow.
Is that Pelagothuria?
Oh,
nope, it's a Medusa. Sorry. It's a really neat Medusa, though.
Is that polyp eating?
No, that's a different one.
Different one?
Yeah. that polyp-eating medusa, it seems that we only find it on the coral. It's very difficult to find on the water column.
Okay.
I would imagine it's so tiny, it's maybe a centimeter, and it's red.
Yeah, I think just where we're sitting down, there's not really a suitable rock, unfortunately.
Roger.
And for those at home,
Just I want to land on those sponges.
Yeah, of course. Yeah, that's going to be a tricky dance here.
the camera you all been talking about.
Maybe over here to our left.
Channel 3.
Do you want to put on the telestrator?
Thanks, Jeff.
Beth, did you want to put on the telestrator?
Like some of these maybe
Roger that.
might be loose.
It's a little something sitting on one of those rocks.
Oh,
Nice. A little something.
Park between a sponge and a hard place, as they say.
So soft in a hard place?
So soft and a hard place.
Seems like an easy situation to get out of.
It's
All
true.
right. And can we get a partial zoom on this?
Yeah, one moment there. Okay. Go ahead and push on in there, please. You looking at the stalk of the old sponge there?
Kind of looks like it. That piece was probably too big to pick up though, right? It's like 20, 30 centimeters.
Yeah, that's pretty large, that one there.
Okay. Can we zoom back out, please?
Don't want to give Steve another 50 pounds to ship.
No.
I don't know. Yeah, I mean,
Can we get a partial zoom on these pieces here?
I didn't know we were going to
Yeah,
be
sure
shipping
thing.
half the
Go ahead, Jeff.
seafloor of the Pacific Ocean across the country.
Just the heaviest parts.
Do we still have the pool as to who's going to get closest to the total amount that we ship? Or did we give that up? Because Steve would be winning at this
This
point.
one might be good. Can we try and see if we can get that one?
Yeah, should they?
Yep, there it is. Stills cam is on channel 3 now. For folks who are interested.
Oh, we can see it. Thanks, Jeff.
Spicy.
It doesn't. It's not full screen because I have a couple of other processes running on that computer, but you can get an
Right, pull
idea
right
of what
there,
it looks
Jeff.
like, the interface. Yep.
And from my understanding, we have some floaty bits in starboard bio. Is that correct?
Yeah.
This would be going in the forward bio.
Oh, okay. Well, that makes our life pretty
Yeah,
easy.
starboard bio does have floaty bits, but microbes in the front, sponges in the side.
Microbes in the front, Raj.
It's like a mullet.
Our watch
So
slogan.
if you're looking at channel 3, this is an
Sorry?
example of one of these still cam photos. That we just opened up here.
Nice. And was that manually taken or was that on automatic?
This one was actually automatic.
Nice. Well done, camera.
Yeah. So there's a little bit of the shroud of the camera here in the corner, but we can lop that off and
I'm thinking a
We
Dremel tool can
edit
take
the
care of
photo.
that.
All right, go ahead and push on in there, but please, Jeff. You want the elongated one on the bottom frame, right?
Yes.
Okay.
So for our viewers at home, as I mentioned, there's many objectives of our expedition here in the Papahanaumokuakea Marine National Monument. And one of them is to understand the alteration of the rocks that are on the seafloor, as well as the microbes that are resident in these rocks, and how they connect to the microbial communities in the water and the surrounding sediment. So the sample that we're going to be collecting here will be for that part of the project.
Surprisingly stuck on there.
Yeah.
Huh.
It looks like the one right on top budged a little
Not
when you
moving.
nudged it.
Why'd you go figure that?
Come on, little rock. You want to come with us, don't you?
with the beans.
Seafloor is full of surprises.
All right. This
That
rock is
guy.
not as loose as it looks.
No.
Well, I think we have some other good candidates here. Maybe the rounder one first, and then if that
Sure
doesn't
thing.
work, the angular one.